Okay, all right, welcome back. So the next lecture, it will be about research methods in decision neuroscience. And before we talk about decision neuroscience, the methods, we also talk about what decision neuroscience means, and then we talk about how we discuss, how we investigate decision neuroscience, decision making, the neuroscience of decision making. I'm already talking about that, okay? Good. So decision neuroscience warm up, experimental design, and the statistical inference, right? So I hope you do learn something important for your own study. So the first question is, what is decision neuroscience? So here, this is a phrase, decision neuroscience, two words. One is decision making, decision, the other one is neuroscience. So decision making or decision neuroscience is basically the neuroscience of decision making. And sometimes if you see in the literature, it is also interchangeable with another term which is called neuroeconomics, because in economics, the economists, those researchers in economics, they also study decision making, even though from a slightly different perspective. So neuroeconomics or decision neuroscience. So here there is a research article from not too long ago. It basically says it is a consilience of brain and decision. Here there are two statements. One is that economics, psychology, and neuroscience are converging today into a single unified discipline with the ultimate aim to provide a single general theory of human behavior. So then this unified thing is called neuroeconomics. So the goal of this single unified discipline is to understand the processes that connect sensation and action by revealing the neurobiological mechanisms by which decisions are made, okay? So it basically tells us it uses neuroscientific tools to study behavior, to study decision making, to study action, right? And also the psychological part, for example. So as I said, because there are two parts, one is decision part, decision making, the other one is the neuroscience part, the neuroscience. I will only use, like, for example, 1 or 2 slides to introduce each of the two domains, especially the neuroscience part, because this is not the focus of the course. I mean, introducing which brain area is named, so how they are connected, it is not the purpose of this seminar. If you're interested in that, then neuroanatomy might be the course that you have to take, okay? So neuroscience is basically the scientific study of the nerve system, and it is a branch of biology, okay? So we have the neurons, we have the dendrites, and those are the things that you might have to learn from another course, not from here. So there are some connections, there are synapses, and then here on the surface of the brain cortices, and there are some different areas, for example, the motor area and the visual area, and also midbrain, right? Some striatal brain areas. So those are different names. There are like many names of the brain. So if we talk about that, then we will have to take, as I said, another semester, because then that's the course for anatomy. So for now, what we understand here is as simple as knowing neuroscience is the scientific study to study the brain and also the extended nerve system, because the spinal cord is also part of the nerve system, right? Not only the brain. Okay, anyway, basically in this course, because we do not really look at spinal cord for studying decision making, so we could think about that we only look at the brain, okay? So the science to the scientific field to look at the brain, neuroscience. Let's put it this way. And we might want to talk, spend a little bit more time to talk about decision theory, because this is important. So this is the content, the core parts of the seminar, the decision part. We use neuroscientific tools to study decision making, not the other way around. We do not use decision making tools to study the brain. So we do not do this, right? So then it's very important to understand what decision making is. So the underlying field or theory, it is called decision theory. So apparently it is a theory about decision making. So there are a few examples I can give you right now. So the first, so do I see red color or the green color of the traffic lights? So whichever you see, it depends on which signal goes into your eye. So that's the sensory system. So go to your eye and then go to your visual cortex, right? Another example is, shall I bring the umbrella tomorrow? So this is also a decision. So this depends on something which I do not know, namely whether it will rain or not. It depends on your prediction. So you look at your forecast and then to see what is the probability, the chance that tomorrow there will be a rain, and then you will act accordingly relative to the prediction. The third example, am I going to smoke the next cigarette? So here, for example, if I make the same decision sufficiently many times, it will kill me. So supposedly I already smoked 10 cigarettes. Am I going to smoke the next, right? So this depends a lot on how many cigarettes I have smoked previously. And then I will make the decision. The last one is the committee has to make a decision, but its members have different opinions. And then how to overcome disagreement here is quite important. So assuming that there is a PhD defense committee. So the PhD candidate is presenting his or her own research, and then there are 3 committee members. 2 says give a 1, 1 says give a 3. So then you see there's a difference, right? So it is the responsibility of the committee members to resolve disagreements, and then they have to give 1 score instead of 3, instead of giving the average, calculating the average. So the main assumptions of decision theory is that there are options to choose from. So recall this example. If I want to go to eat somewhere, I have options to choose from. I don't have to always go to 1. If I do, then that's because I want to. It's not because there is only 1 option. There are multiple options to choose from. Second, so we choose in a non-random way. So our behavior is not random. We do not behave randomly. Recall this slot machine example. If this slot machine is on average always giving more positive outcome relative to negative outcome, so on average this machine is very good, we want to play with the machine, then we play with it. If this machine is a bad machine, it returns monetary loss more than monetary gain, then we might not want to play that anymore after a few trials and errors. On the contrary, if we behave randomly, so every time we see this machine, even though we know the good or the bad of the machine, if we behave very randomly, we always behave 50-50, so chance level. So maybe out of 100 times, we play 50 times, right? If this is a good machine, out of 100 times, maybe we play 80 times. If this is a bad machine, maybe out of 100 times, we play 20 or maybe even less, even 10 times, for example. So we choose in a non-random way. Another aspect, it is also something we mentioned already the last week. So our choices are goal-directed activities. Again, the same example. Both are good choice options. Both are interesting. Both are nice. Let's also assume the smoothie also tastes good. Both also taste good. But which one you will choose really depends on your goal. Your goal, is it to reward or to treat yourself, or maybe you are on a diet, you want to stay healthy, you do not want to gain some weight, maybe here. So the decision really depends on your goal, okay? So it is called goal-directed activities. So if you combine the three properties together, first, there are options to choose from. The second, we choose in a non-random way. The third, our choices are goal-directed activities. So if we combine all the three above together, we could say overall decision theory is concerned with goal-directed behavior in the presence of options, okay? So putting everything together, decision, we talk about neuroscience, it is science to study the brain, and we talk about decision making to study goal-directed behavior with multiple choice alternatives. So putting together decision neuroscience or neuroeconomics, there is a core part of the neuroeconomics. There is also a extended parts of the neuroeconomics. The core parts of neuroeconomics or decision neuroscience, it is a field of neuroscience that studies the process connecting sensation and action by revealing the neurobiological mechanisms by which decisions are made. This is repeating what I said already from that research article. So this extended neuroeconomics, it is a multidisciplinary field that studies mechanisms of decision making by integrating the evolutionary, neurobiological, and social approaches. So it is quite a interdisciplinary and also multidisciplinary field that tries to connect together different fields like psychology, economics, neuroscience, and social science, for instance, also. So it's pretty interesting. That's why I'm trying to bring this topic to you, because I find this at least a very exciting topic. So the decision neuroscience or neuroeconomics, it has one goal, but with two perspectives. So the goal of neuroeconomics is an algorithmic description of the human machine for choice. Okay, this is said by this the author of that paper, and this is also the author of the neuroeconomics book, Paul Glincher. So here we talk about this algorithmic description. So what that is, this is basically computational modeling, right? From the last lecture. So there is a question about, is this common in research? I can't really speak for the other field, but as I said, at least for decision neuroscience, decision making using computational modeling is very often, and research studies using this method have achieved a lot of interesting achievements and findings to have a better understanding of how the brain, how people make decisions. So this is the one goal, and different perspectives. So here, this field, it combines experimental techniques from neuroscience, psychology, experimental economics. So here, those methods, they include electrophysiology, fMRI, eye tracking, behavioral measurements, and also, again, here, computational models, right, from neuroscience and also from economics. So the perspectives are like twofold. So one is from maybe more experimental parts, the other one is from a little bit more computational parts. Okay, good. So one goal, two perspective. And some questions can be asked some questions can be studied in the field of decision neuroscience. For example, so what are the computational processes that the brain uses to make economic decisions? What is the neural basis of these processes? How and where are values and probabilities combined in the brain to provide a utility signal? So how do emotion and cognition interact to shape decision? So what are the mechanisms of social decision making? And so on and so forth. So all those questions can be asked in the field of decision neuroscience. Okay? So imagine we could look at here. So how do emotion and cognition interact to shape decisions? So this is an idea. So I'm trying to connect what we have discussed from the last lecture to this one. So this is an idea. What we talked about the last lecture, we talked about how to translate, how to form or formulate hypotheses from ideas. So this is the idea. Okay, good. How do emotion and cognition interact to shape decisions? Hypothesis, we can be a little bit more specific, right? This is the first step. We need to be specific. We can use something similar to the example we have, even though that might be stupid a little bit. So we can say positive mood states by watching, for example, funny events, the same idea, could increase the risk-seeking behavior in the decision-making task. I'm not sure whether this is a valid question, but it can be answered, right? We can let them watch videos. One is funny, the other one is ordinary. So both before and after, they will be asked to rate how do you feel, how I feel. And then this is the manipulation check. Very good. We have covered this. And then after that, we show them a risky decision-making task. And then we compare the percentage of the risky choice taken from the participants. And then we could compare how this emotional mood state might affect risky seeking behavior. Okay, make sense? Good. So we talk about study design, and then let's then in this lecture talk about the techniques. Okay, because if you recall from here, one goal to understand the neuroscience of decision making from two perspectives. So here, the study design I talked to you, the seemingly stupid example I gave to you, it's from this perspective. And then kind of from this perspective. And then there are some other perspective from here. And then I'm also showing you later in this lecture some some experimental methods that can be used to study decision making. Okay. Good. So before we go forward, any questions, comments regarding here, this short introduction of decision neuroscience? Everything's all right? Okay, good. If no more questions, so let's start this part, the research methods in decision neuroscience. The very first, for example, if we study decision making, we study decision making, right? So in the laboratory experiments, we invite participants to come into the lab, and then we ask them, if you see this, for example, press left, and then if you see something else, press right arrow key on the keyboard. So those are the decisions, the responses, the dependent variable we could observe from these experiments. Okay, so what is that? What that is? That is behavioral measurements. Okay, behavioral measurements, behavior measures, button press, for example. There's IQ score test. their proportion of making risky decision making, so their learning performance. So all those can be classified as behavioral measures. So last year, not too long ago, a few months ago, so there is a special issue in this journal called Neuron. So Neuron is a journal that publishes in general neuroscience studies, also including sometimes decision-making studies, like also including memory and single cell recording, a lot of stuff in neuroscience generally, generally speaking, they published that study. So last year there was a special issue. So they focused on behavior. The entire issue, the articles published there, they talk about behaviors. They use behavior as a window to look at the brain, okay? You might wonder why neuroscientists, they also look at behavior. It is because for them, behavior is the readout. It is the outcome that you can observe from either human or animal. So that is the window to look at the brain, okay? So this is when this special issue was coming out. This is on Twitter. So the official accounts of Neuron, they have a claim. They say behavior matters. So they say, even though it is a neuroscience journal, they really look at behavior. Behaviors, they do matter, okay? This is the claim. So this special issue is all about behavior, from rodents to humans to neural circuits, from to psychophysics, to exploring learning, attention, decision-making, and a lot of thoughts and reviews, perspective, primers. That is very interesting. And I would also like to recommend you read some of the papers if you find this interesting. There is the link you can press. So go to that special issue, find maybe just one or two papers that you're interested in, and look at how behavior is studied modernly, like from last year, right? How decision-making or in general behavior is studied by neuroscientists. And she is the senior editor of Neuron. So it is her effort that puts all together those papers, those articles, as in this special issue. So she was also pretty happy that she put together this special issue. It has been a lot of fun and it's pretty interesting. And here there are some subjects that is studied in those papers, human and animals, for example. The idea, what I want to convey is that, say, animal behavior can be produced intentionally to achieve positive outcomes. So as illustrated here, the cover. So the initiation of action, such as the approach of a thirsty mouse to a water dispenser, is a powerful signal to the rest of the brain that a reward is imminent. Okay, so here there's a mouse. Maybe here this is like too artistic. So here there is a device that the juice could drop from the device so that the mouse could drink from it. So this is a highly motivational state because the mouse is thirsty and the mouse is trying to get the reward, not monetary, just primary reward, food and water. This is very important to look at the behavior. So now I want to ask you, for example, in a real laboratory environment, what behaviors can be measured? So think about it. I give you one example, choice preference. So we could ask participants to give a binary response. Say there is a risky gamble. And we ask participants, do you want to play this risky gamble? Do you want to take the risk? Yes or no? Okay, binary. It can also be a continuous, a subjective rating. So on a 1 to 9-point scale, how much do you want to invest to play some game, for example? So this is also kind of decision making. So those are responses that we can record from participants. So now, please type in the chat box to basically continue the list. What else do you think can be measured as behaviors in the laboratory settings when we study psychology, for example, or decision making in general. I see a few responses: eye movements, physical reactions, the time it takes for a subject to decide. Yes, everything's good. And everything else? Duration of the behavior. Yeah, the same, similar to reaction time. Yes. And what else? Intensity of the behavior. So just recall the experiment you have participated from last year, from maybe even before. So imagine what's response you have to give to that particular experiment. And then those are basically the measures, right? Hormone level, cortisol, yes. We will talk about that. Yeah, blood pressure, yes, okay. Dopamine level, skin conductance, yeah, good idea. Social influence. Okay, good. Heart rate, yes, okay. Great, thanks. So many of the responses you gave, they fall into this category, behavioral measures. But actually they fall into another category, which I will tell you in just a minute. So here, let me just complete the list I have. So we could record choice preference, ratings, yes or no answers. We could also have confidence ratings, right? So if I make a decision, if I say yes or no to a risky gamble, do I want to play a risky gamble? If I say yes, maybe the program will ask me how sure I am the gamble will release a positive monetary reward. And then I might say, okay, sure, maybe somewhat unsure, somewhat sure, not really 50%, or maybe just unsure. So confidence rating can also be very important to record from experiments. Reaction time, so some of you have already talked about that in your responses. Thank you very much. So reaction time, basically how long that takes for participants to make that action after they have seen the stimulus on the screen, okay? So we could also study process tracking. So eye tracking, it is something you mentioned. Some of you mentioned in the button box, in the chat box. So eye tracking, and there's something else also. So one is called mouse tracking. Another one is called eye foot tracking. Okay, I show you one example. So here, one eye tracking is basically you put an eye tracking device onto the head of the participant, and then to measure the gaze pattern. So if here, this is a screen, we could measure how much time the participant is spending on the brownie versus how much time the participant is spending on the smoothie. So the eye is spending, okay? And also we could also measure not only the time at each point, we could also measure the jump, okay, from here to here within this option, and also between this option, how much time they look at back and forth before they really make the button to make the choice. Do I want the brownie or do I want the smoothie, okay? So the mouse tracking looks like this here down below. Let's say this is a screen of the computer in the laboratory. Here, the mouse always starts from here, okay? If the participant is really eager to make a choice to choose the broccoli, then there's a straight line. But maybe the participant is a little bit hesitant, and he or she wants to have the brownie, but maybe then he changed his mind, maybe then changed eventually to the broccoli. Okay, so the process tracking here, you see the curvature, either very curved or straight, is a process. It reflects some kind of thinking before the participant is making the choice. It gives us much richer data relative to only a yes or no choice, right? Only left or right. With the process tracking, we know more about what might be happening in their mind. So one of you talked about physical measurements. So here it can be an effort tracking. So here, for example, I have a squeeze bar. I can squeeze it. And if I squeeze it, the blue color will go up. And if I do not squeeze it as hard as I can, maybe the blue color is here. So the experiment can be that if I want to have the brownie, and then I need to make a threshold. I need to reach a threshold so that I will be rewarded a brownie. So this is the screen. I am a participant. The experiment program shows me the brownie and then asks me, if you reach this threshold, the yellow bar, by pressing this squeeze bar, and then the effort will increase, and hold it for 3 seconds. And only after that, I am able to consume the brownie. So otherwise, no, not possible. So this is a way to study physical effort. This basically means if this is a very attractive food option, I might be very willing to put the effort so that even though that's very hard, I can still press it, I can still hold it for 3 seconds. And maybe for another food option, I don't know what it is, maybe just water. And if I'm not really thirsty, I do not really want to have it, then I might not be willing to put too much effort in order to get the water, which is something I am not in a great need. All right? Good. And then what else? So a few of you mentioned, for example, the heart rate, the skin conductance. They do not fall into the behavioral measure category, but they fall here in this physiological measures category, in the sense that we measure some physiological responses from the body. One is the heart rate, the heartbeat. The other one is, for example, the facial EMG. So to study the tiny small currents that is produced on your face. So when you are doing some emotions, when you are smile, when you have the sad emotion, the facial EMG, supposedly they might differ from one another. So facial EMG, one example is that experimenters, they attach the electrodes onto the faces of the participants and then to measure the small currents, right? Another one is the skin conductance responses, which is also called SCR in the literature. So here, this is one way to measure the skin conductance. So for example, if you are in an experiment and if you receive some stimuli, so let's put this simpler. So sometimes you might get an electronic shock, right? So when you get an electronic shock, you feel pain in the first place, but your skin might also respond a little bit differently relative to the baseline. So here, this is a way to study the pain response when you got the electronic shock. Cortisol level, yes, of course, this can also be measured. So this is one way, for example, to save and then to reserve the saliva. Yeah, and so on and so forth. So then the behavioral part can be measured, the physiological part can be measured, and what else is the neuroimaging part, right? We talk about decision neuroscience. We talked about decision, so here we will talk about the neuroscience part. So I'm sure that many of you have seen this before. So maybe just write in the chat box if you have seen this before, say yes or no, if you have seen this diagram. Okay, I see quite many yes here from the response. Any volunteer, that's turn on your microphone and tell me what you have learned from this before. Anyone? Volunteer?
Hi.
Yeah, go ahead. I can hear you. Thank you.
Okay, so we've discussed this before, and it's also in the chapter that we should have read. I think it's a listing, kind of a graph to show the spatial and the
Temporal? Yeah,
time
temporal. Yeah, yeah.
resolution of different neuroimaging measurement techniques. So as to compare them with each other.
Yeah, and did you talk about any specific example? For example, fMRI?
Yes, fMRI, PET, EEG, MEG.
So, for example, could you describe the property of functional MRI here on this x-axis and also on this y-axis?
So if you would compare fMRI with, for example, wait, the chat is in front of the graph. For example, I can't see EEG.
Here, yes, the ERP is named a little bit differently. Yeah, yeah.
So you would see that the time resolution is a little bit slower. So yeah. EEG has better, like it provides instant information. Yeah. But you can see much more of the brain itself. You see more of the spatial locations.
Yes, because here you see something lower, right? Relative yes. to here, the red. Okay, great. Yeah, thanks. Okay, so this we will see this graph two times. So here is basically overview. You don't have to know which is which, what is what, if you have never seen that before. So we will talk about not everything, but many of the techniques here. And then later, when we have introduced many of them, and we look at here again the paragraph to compare their spatial resolution here, and also the temporal resolution here, okay? So before talking about that, one thing I wanted to talk about is, so whenever you see a graph, because you will read a lot of papers for this seminar, you will see quite a few graphs, figures, images. The very first step as a good practice is always to find out what the axis means. So what the axis mean? So here we have x-axis, the time on the log scale, doesn't matter the log scale. So you see here, this is very short, milliseconds. This is second, minute, hour, and day. So as this number from on the x-axis increases from low to high, it basically means it becomes less precise. Milliseconds is very precise and it goes to a second level, then maybe it's a little bit less precise, and then day is the least precise temporal resolution. So the similar idea goes also to the y-axis. It is the spatial resolution. The unit is millimeter. So here it is also on the log space, doesn't matter. So we have the brain. This is very broad. This is large. It's not very precise spatial resolution. So the map, the column, the layer, the neuron, the dendrite, the synapse. So here is very tiny, right? There are some techniques, not really here, nothing here, but there are some techniques could study the dendrites. For example, here the light microscopy that could look at the brain at a very precise spatial resolution. Good. So then let's see quite a few of, on this graph, quite a few of very often used neuroimaging techniques to study decision making. So the first one is called a single neuron recording. It is used not so often on humans. So it is used quite often on animals, monkey or mice. So it is very often. So in this simple experiment, let's say there is a mouse. So there is a cue, and then the mouse is supposed to learn something, to expect, to predict some reward, for example, juice from the environment. Okay. So here, this is the diagram of the results of a single neuron recording. So each line here, each row, each line is one neuron. So even though that's called single unit recording, that doesn't mean only to record one neuron. It uses kind of a small device, very small, maybe a few millimeter by a few millimeter, maybe centimeter, depends on the size. So the unit could record multiple neurons under this device. So each line here is one neuron. One neuron, one line, second neuron, third line, third neuron, fourth line, fourth line, fourth neuron, okay? So if there's a firing, you know the neurons, they fire, right? If there's a firing, then it is marked in black. For example, here, for example, here, okay? If the neuron doesn't fire, it just stays there at the baseline level, and then it's empty. So it's white, in the white space, okay? So at the very top, it is the summary, the cumulative sum of how many neurons are firing at each time point. So the x-axis basically means time. So here, let's say if this bar is long, that means at this time point there are quite a few firing neurons, okay? If in this case, you see there is on average, the total sum, it is not much. Then you go down, you didn't really find this column. There is a firing neuron, maybe one or maybe zero. Okay, it's quite small, not easy to see, but basically here the neuron doesn't really fire. Okay, good. This is the diagram, how to read it. So here, this is the mouse. So when they see a cue, they would receive a juice. Okay, so immediately after the time the mouse is receiving the reward, the juice reward, the juice drop, then the neuron that is recorded, they fire. There's a huge fire, right? Cumulatively. So they fire and then they go back to the baseline. So the general idea of this diagram is that this is time. This is the time point when they get the juice reward. So nothing going on, not much going on. And then, bam, gets the reward. And then there's a huge increase of the neuron firing rates and then drop back to the baseline. Okay, so this is the how to read it. Good. So this experiment is very interesting in the sense that because there is a cue, okay, there's a cue, then some cue is like Pavlovian conditioning. So if the cue is associated with the reward, so then if they see this cue again, even though they do not yet see the actual reward, they might already have a very high expectation, right? Because they associate the cue with the potential reward. Because they're linked, because of training, they think whenever I see this cue, there will be a reward. So the neuron firing rate is high. So the time it fires becomes earlier relative to the actual reward is delivered. They haven't seen reward here. When they see the cue, they are already very excited. They're already very excited by seeing the cue, for instance. And when they see the cue and when they really get the reward, they are not really surprised, because I know if I'm a mouse, for example, I know if I see this cue, I will get a reward. When I see the cue, I'm already excited. I'm excited enough. And then later after, if I really see the reward, I know it. There's no surprising. There's nothing really unexpected. So the actual reward delivery really doesn't induce so much neuron firing rate. Okay, so here, this is the important part. And which is even more interesting is that because there's the cue, this cue is previously associated with a Joule's reward. And then at the later part of the experiment, we know, okay, there is the cue, then the mouse is seeing the cue, then there's some neuron firing. But if there's no actual reward delivered, then the neuron you see here, they stop firing. So then here, it means they are surprised. So the intuition is that I see a cue, I know a reward is very likely to happen. But what is really happening is that, very sad, there is no reward. So there is a huge surprise signal, okay, here in this case. So in the literature, this is called the reward prediction signal, the reward prediction error signal. So reward prediction error. They predict something positive, but they receive nothing. So then their reward prediction error is negative. So here, they predict positive, they receive positive. The prediction error is 0. They're not surprised. And then here in the beginning, at the beginning of the learning, they haven't yet associated the cue with the reward. They have no expectation, but they receive something very positive. So then here, this is a positive reward prediction error, okay? So then if you read the paper, you will find out that this is a dopamine neuron. The dopamine neuron is encoding, is really associated with the firing of the reward prediction error signal. And this is the main finding of the paper. So believe it or not, this paper has been cited for nearly 8,000 times. This is the most important literature in the field in decision-making, in learning, and in decision neuroscience, okay? This is very short, maybe I think only 4 pages, I forgot, only 4 pages. And if you're interested in, so take a look. And this graph you will see very often in the field of decision neuroscience. So we spent quite some time explaining the experiments of the previous mouse study, and this one I can go a little bit quicker. So here, there is a monkey, same idea, to record directly the neural activity from the brain of the animal. And let's see, there is a perceptual decision-making task. So the animal has to choose left or choose right. Depends on how much coherent the color is. For example, if in this middle part, there are more red colors, then the decision is here. If there are more green colors, then the decision is here. And then this study basically studies the reaction time and also the trajectory, how the animal behaves in terms of making the perceptual decisions, okay? The task here is quite interesting also, but the point is basically to show you how a single neuron recording can be used for decision-making studies. Good. A summary about this technique, single neuron recording. So it is the only direct measure of neuronal activity. So we study the fMRI, we study the EEG, we study something else. So all the others, it is an indirect measure of neuronal activity. But here, so the device I talked about to record single neurons, they are attached to the cortex directly. So it is the direct measurement of neuronal activity, okay? So this device sometimes called, this device, kind of a matrix, rectangular shape, it is just called a matrix, the matrix to record single neural activities. It is invasive, yes, because it is direct, it is attached to the cortex, so it needs some operation. So if you see some animal literature, so usually there's a electrode or this matrix, the researchers first have to do an operation to implement this matrix to record the neurons, and then later to let the animal make decisions in experiments. Yeah, so because the matrix is quite small, maybe a few millimeter by millimeter, centimeter by centimeter, it's small basically. So it's limited to only a few neurons. It's not very easy to record the entire neuron from the whole brain of the animal. So it's quite targeted. You need to have a strong hypothesis, right? Because if you only when you have strong hypothesis, you know where to record, and then you can record. But what if you have a wrong hypothesis? You record somewhere, but actually somewhere else is firing. So this might have some problems. Okay, so it can also be unclear information codes. Anyway, there is a review paper. So here. So to talk about the methodology, so single trial neuron dynamics are dominated by richly varied movements. So this is a very interesting study that uses single neuron recording to study the movements during experiments. Some also spontaneous movement. This is also very, very interesting to read. The paper is from last year, I think. The second technique I want to talk about is brain lesions. So somewhere of the brain is damaged. So this is called brain lesions. So for example, in one study from quite a few years ago, from 1994 by Rolf Adolphs, and he and the entire research group look at the behavior from a amygdala lesion patients. So you'll see here, this is the brain of a healthy participant. Here with the white triangular shape, it points to the bilateral amygdala. So this is how the amygdala looks like in the healthy participants. And here, this is how the patient's brain looks like. The damage, the lesion of the bilateral amygdala, okay? So if you have learned or heard about the brain area called amygdala, so you might know that amygdala is found to be associated with emotion very often. So then this research does study the emotion recognition of the patients relative to the healthy control. Okay, see here, this SM is the name, the code for the patients, here for healthy control. So again, this is a graph. What we do is we look at the axis. Here, x-axis shows the facial expression, the happy, surprised, afraid, angry, disgust, and sad, okay? And the y-axis means the mean rating of all faces in an emotion category. It's basically to ask the participants to categorize the faces. So what is the accuracy then? So you see here, on average, it is always that the circle here, the circle, is higher than the square, not square, sorry, the triangle, so the black triangle. It's basically that's the ability to recognize surprise, for example, afraid and angry, disgust, right, and sad. The ability to recognize those faces, it's quite reduced in these patients, okay, especially when these emotions are negative. For happy, you see here, maybe there's not too much difference between the healthy control, the circle, and the triangle, the patients. So anyway, this is brain lesion study. We could compare some aspect of the behavior in between the patients and the healthy control. And also here, there's a combined behavior together with eye tracking study. You see here, the healthy people, they look at the eyes more often as a cue to recognize emotions, whereas the patients, they kind of avoid looking at the eyes. They look at more a little bit down on the face, nose, the area. So here, that makes it difficult to recognize the corresponding emotions. So brain lesions, it is rare. So if we want to study amygdala damaged patients, there are not too many in the world. So here, this study, if I remembered correctly, there is only one patient in the paper, and maybe two, relative to a few healthy controls. We cannot say we recruited 20 amygdala lesion patients. This is not really possible. Okay. In South Africa, there's an area. There are quite a few Uberhuter patients. They are basically amygdala damaged patients. So usually, people there, if you want to do brain lesion studies focusing on amygdala, you have to travel to South Africa, because there you find the most dense population of amygdala patients, amygdala lesion patients, damaged area. Okay, so even though it's not so easy to recruit a lot, maybe 5, 6. This is a large number of brain lesion patients, so it is very rare. There's a question, why in Africa? Is there a reason? I have no idea about this. Sorry about that. So brain lesions here, the consequences are long-term. So as long as the brain is damaged, it is damaged, right, throughout the rest of the life. So the consequence is long-term. Even though it sometimes can be compensated by brain plasticity. And this is also a very good tool or technique to study the causal role of the brain. So if we know from this example, we know that in healthy control, they could recognize the emotions relatively good, but for the patients, the recognition behavior is a little bit reduced. And we know the difference between here, the patients and the control, is that there is a difference in amygdala, the brain area. So we know causally here, the amygdala is more like the reduction or the lesion in amygdala is causing the failure in recognizing those phases or emotions. So it is to show the causal role of the brain region. And then to bring us some unique insights about the brain. So here, the same author, on the original paper, Naraf, and he wrote a review article 3-4 years ago about human lesion studies in the 21st century. So if you are interested in this particular technique, really recommend reading that paper. So the next one is called Transcranial magnetic stimulation, so TMS. So we use a coil, right, to disturb the brain, the activity of the brain. So we know that magnetic field could generate some, here this is magnetic field. So the magnetic field could generate some currents, and then this could affect, for most of the cases, to disturb the brain activity. Okay, so this is the general general take of this technique. So we could temporarily downregulate some activity of a particular area of the brain. Okay, so look at this graph. So this is, let me give you a little bit background. So there is a social decision-making task. There are some unfair offers. So the question to the participants is, do you want to accept this unfair offer or to reject those unfair offer, right? And then from a previous study, they know that there is a specific area here, dorsolateral prefrontal cortex, might be related to this kind of behavior, the acceptance rate. So then the research team, they stimulated the left and also the right part of the dorsolateral prefrontal cortex, so bilateral. They stimulated here, they stimulated here, and sham means a control condition. So what can be seen here? Only stimulating the right part of the dorsolateral prefrontal cortex will increase the acceptance rate, whereas stimulating the left one does not increase relative to the control condition. Okay. So there are quite a lot of more details in the paper, but the general idea is to temporarily downregulating the brain activity of a particular area using this technique. There are some other brain techniques, brain stimulation techniques. This one is called transcranial direct current stimulation, called TDCS. So here, a short introduction of this research is that there is a research team. They are from Zurich, I think. So they have a group decision-making task, and all of them are stimulated using this TDCS device. So what is different from the TDCS and TMS is that the TMS, you usually use it to downregulate the activity of the brain. If you use TDCS, it goes bidirectional. It can go up, it can also go down, okay? So I only show you here one example. Sham again means the control, and anodal and the cathode, one means up, the other one means down. And you see here there is a trend, right? So there is a very nice effect of stimulating the brain area of the social decision-making task. So I will try not to talk about this too much because this will be also a paper that some of you will present during your presentation. So just show very briefly the idea of this brain stimulation technique. The experiment and then the funding, we can wait a little bit to see how the participant will present to us. So taking TMS and TDCS together, so both of them, they are non-invasive brain stimulation. We do not have to do operation to stimulate the brain, right? So TMS uses magnetic field, TDCS uses the current, right? The currents. So before using that, if you're interested in this non-invasive brain stimulation technique, you will have to decide what time resolution you want to stimulate. So you want to stimulate. Is it transient or is it long-term effect? How long do you want the effect to last? So it depends on the time duration. The technique can be adjusted accordingly to accommodate short or long effects. Short means maybe a few seconds. Long maybe means half an hour or so. Not really days. It's a few minutes 30 minutes is quite long already in these non-invasive brain stimulation experiments. So using brain stimulation, similar to the brain lesion techniques, it also shows some causal neural mechanisms. Some limitation is that both of the TMS and the TDCS, it couldn't reach too far from the surface, from the cortex of the brain. So it is quite difficult to stimulate deep brain areas in the midbrain, for example. And so both of the techniques, they are quite loud. So it's quite, for some participants, it's not comfortable to listen to those sounds and noises and muscle constructions at the same time when they are doing the experiment. Okay, those are things to be considered. So again, if you're interested in this non-invasive technique, there is a review paper studying and modifying brain function with non-invasive brain stimulation. This is also very recent, I think from the beginning of 2019, last year. Good. So now we can talk about EEG and MEG. So EEG is the recording of electrical activity along scalp produced by the firing of neurons within the brain. Okay, so why am I talking, and also why people usually are talking EEG and MEG altogether? It is because they are similar, but they have differences. So here, what I show in this red color, that's the difference. So EEG measures the electrical activity. So the MEG, by the name you can see here, the magnetic, so it measures the magnetic fields produced by the electrical activity in the brain. So we know that there are neurons in the brain, the neurons, they fire, and the neurons, if they fire, they produce electrical activity. So the EEG, they directly measure the electrical activity generated by the brain. And we also know that if there is electrical field, and that it also generates a small magnetic field, right? So that's from physics, we also know that. So here from MEG, it measures the magnetic fields produced by the small currents, the electrical activity in the brain. Okay, good. So here there is also a directional difference between EEG and MEG, but this requires you know a little bit physics to understand the difference. Not very important here, but you know here this is important to differentiate two methods. One is to measure the electrical activity. The other one is to measure the magnetic fields produced by those electrical activity. Okay. So let's look at a simple experiment. Here there are two choice alternatives. One is 25, the other is 5. So then the participant, they have to make a response, and then they will see the outcome. Okay, green means gain and red means loss. Okay, details go to this paper and then to see how this is done. So this will also be a paper for the presentation, so I don't want to talk too much about that. So what can be seen here by using an EEG experiment, there are two conditions. One is gain, the other one is loss. So you see here, after it's around 250 milliseconds after the stimulus onset, there is a difference between the gain and then the loss. Here, then this electronic activity is recorded from this area of the brain. Okay, so you can use EEG to study how the brain is responding to gain outcomes versus loss. Here, gain outcomes versus loss outcomes. So the device of EEG looks like this. MEG is pretty large, honestly. So here, this is the MEG machine. It's quite expensive also. So there's a comparison between EEG and MEG. Signal magnitude here, so large signal is to detect. For MEG, the signal is quite tiny, honestly, and it is a little bit difficult to detect. That's why it requires such a large machine to detect those tiny signals, the tiny magnetic fields produced by the electronic activities of the brain. EEG is relatively cheap, and the MEG is rather expensive. And there are some other aspects that you might also want to pay attention to. So here is important, the temporal resolution, both of them, they're very high, okay? And spatial resolution, here the MEG wins over the EEG. So it has both a high temporal resolution, at the same time it has a high spatial resolution. So it could go to the unit of 1 millimeter. So here it's 1 centimeter, right? This one is 10 times more precise relative to this. Good. And then there are some other aspects you can also look at. Okay, the next brain imaging technique is the functional magnetic resonance imaging, the fMRI machine. So here, this is the machine. It's also a huge machine. Participants, they lay down in the machine, and then this machine is measuring some activity, okay, of the brain when participants are doing experiments, for example, decision making. So one thing to pay really attention to is that the MRI machine does not measure brain activity. It does not measure electrical activity. It does not measure the magnetic fields, okay? So EEG, we know it measures electronical activity. MEG, it measures magnetic fields. MRI doesn't do either of them. So what does that measure instead? Instead, it measures something called a bold signal. It is a blood oxygenation level dependence, which is used to detect brain activity, okay? So the idea is that when somewhere of the brain, when some area of the brain is more activated, then there are more oxygen, because if the brain is activated, it requires energy. It consumes energy, and energy comes from oxygen. If the brain is in a resting state, not too much energy is needed by the brain. So then in this case, the oxygen level is not much. So what is compared is how much change from this resting state when there's not much oxygen need to be consumed to here when a lot of more oxygen has to be consumed if the brain is activated, okay? So this machine measures here the bold signal. And we use that as an indirect measure of the brain. So if we observe a higher bold signal, we know that the brain is more activated because more oxygen is needed. And if we do not observe a high bold signal, then that means the brain is not activated, using the evidence that the brain doesn't require that much oxygen during one experiment. Okay, this is the idea. Good. So one example could be that the participants, they see food choices, and then the experiment asks the participants, how much money do you want to bet? And then this is the feedback of the experiments. And then they go on and go forth. And then here we could see that an area in the brain, so it's a median OFC. And also here the DLPFC, those two areas are related, for example, to gain. I'm not very sure, and also because this is a paper, I do not want to talk too much about that, okay? But this is the typical image you will see from fMRI studies. You see in the three-dimensional illustration, X, Y, and Z, so that's the location, the area of the brain. In this case, here, this one, this one, this one, they are the same area. So showing from different perspectives. So this is a better illustration to show precisely where the activation is, okay? So here the same idea, this activation here and here, they are all the same thing, just shown from different perspectives. MRI technique, it is also non-invasive. It has a good three-dimensional resolution. So I showed you the same activity shown from three perspectives, 3 dimensions. It is an indirect measure. Pay attention, really remember that it doesn't measure the brain electronic activity. It doesn't measure the magnetic fields produced by the brain. It measures the bold response, the oxygen, the blood oxygen. So the timing is usually not very good. So the timing resolution, the temporal resolution is a few seconds. So if you want to really look at the temporal resolution of the brain, MRI might not be the good choice for you. And it is pretty noisy, the same as MEG, it's pretty loud. So when the machine is turning on, when the machine is turned on, if you have experience before, it is pretty loud. Even though the experimenter, they give you the ear plugins, it can still be very loud and annoying to listen to. And also it has some restrictions for participants. For example, if you have tattoos around your neck, if you have piercings, then it's not recommended to go into the fMRI scanner. And again, because we showed quite a few review papers per each technique, similar idea, there is also a review paper regarding the functional neuroimaging methods. So the authors, all of them are like the leading experts in this neuroimaging field. Good. Now again, the second time of the same diagram. So the x-axis, the temporal resolution, the y-axis, the spatial resolution. We talked about this one in the first place, single neuron recording. So it goes down to the neuron level, right? And then the timing, it depends on how much neuron fires. It is a direct measure. It requires operation. So it depends on how the neuron fires. It can be short, it can be long. So it is with the high spatial resolution and a flexible temporal resolution. We talked about also lesion studies. So here, lesion study, it is pretty precise, honestly, but then the time resolution is not very high, okay? We talked about also quite a few brain stimulation techniques. We talked about TMS, we talked about the TDCS. One example is here, the TMS. So the spatial resolution is not pretty high, but the temporal resolution is high. And here, the EEG and MEG together as, what's her name? Lena, I guess, as her mentioned. So EEG and MEG has a precise temporal resolution here and a relatively poor spatial resolution. And functional neuroimaging at the very last, we talk about this. So this spatial resolution is relatively high, it goes to here, right? Millimeters, levels of millimeters. But the temporal resolution is like a few seconds or minutes. Not really minutes, but seconds usually here. Okay, good. So all those techniques are useful, and it really depends on which question you're interested in, and then you choose the technique accordingly, right? So for example, if you're interested in the temporal fluctuation of neurons in some decision-making task, then maybe functional neuroimaging is not the good choice because it's quite bad at temporal resolution. And by contrast, if you're interested in the location, which area is much more involved in one decision-making task relative to the other, then maybe EEG is not a good choice because EEG is a little bit poor at spatial resolution. Okay, so those are the considerations you have to pay attention to when you decide which technique to use. Okay, and there are also a lot of more for research methods when studying decision making. We could also, for example, videotape the behavior in the laboratory. We can take the voices as well. And we can also do pharmacological manipulation. We can use, for example, oxytocin. We can use testosterone, for example, to study participants' risk-taking behavior and pro-social behavior. There's something called a PET scan, and this is not used very often nowadays, so we didn't talk about that. Good. So before we close and before I ask you questions, or maybe you ask me questions, there is a distinction or differentiation between two terms. So one is called measurement, the other one is called manipulation. So what is measurement? Measurement means what we can measure. So we can measure behavior, we can measure choices, we can measure the confidence, we can measure also skin conductance, the heart rate, we can record video, we can record their voices, we can record facial EMG, right? We can record even the single neural activity using EEG, MEG, fMRI, PET scan. So all of them are measurements. So what is manipulations on the other hand? So this manipulation relates to the idea of our last lecture when we were talking about experimental design. So what we want to manipulate: high emotional mood, low emotional mood. This is not measurement. This is something we can induce from experimental manipulation, right? So here, for example, TMS, it is manipulation because we are manipulating some brain area by downregulating, for example, the activity of that area. TDCS as well, because we use stimulation technique to manipulate the area. So invasive stimulation as well, right? And also pharmacological manipulation. The name is there, is a manipulation with drugs, for example, with hormones. Lesions, it is also a kind of manipulation because the brain areas are damaged for those patients. Good. Any questions so far? Anything you are curious about? Anything that you still want to know? Even something that we haven't talked about? Anything related to experimental design and the statistical inference? So jointly from the last and also from this one. Any questions, comments, feedback? Everything's all right? Yeah, there's a question. Will we talk about the chapters you have we have read? You have read? No, not directly, because here I just try to more or less incorporate all those information from the book to the slides. So the book, I will talk about that here. So the book is more or less serves as a supporting material. So I will not really directly talk about each particular chapter of the book. I will not really quote any content of the book, but it's more for you to have a supporting read. If you are interested in here neuroeconomics, or there's another book called Decision Neuroscience, or maybe also this one is pretty famous from Daniel Kahneman, Thinking Fast and Slow. So if you're interested in, you can go ahead and then read it. If you have questions, you can also discuss with me, but I will not really directly and explicitly talk about the book. And also, if you realize, when we were talking about the experimental design and the statistical inference, I was basically trying to synthesize quite a few sources of information from different textbooks. So you can think as that I have already selected the information for you, which I think is more relevant. But you are still more than welcome to read other textbooks to strengthen your understanding. Okay, other questions, comments?